Okay, so welcome everybody, and thank you so much for attending this session of the Brain Science Speaker Series presented by LSU's Multidisciplinary Initiative for Neuroscience Discovery. LSU Mind aims to coordinate and strengthen neuroscience research, education, and community outreach here at LSU and beyond. I'm Susan Duncan, and I serve on the Executive Committee for LSU Mind, along with Melissa Beck, Neela Donovan, and Heather Lucas. We would like to thank the Center for Computation and Technology for funding the Brain Science Speaker Series and providing us with tech support. And we will be hosting another speaker in the spring, so please do stay tuned to the LSU Mind website and sign up on our mailing list. So tonight, for Mind's first ever virtual event, we are very excited to have Dr. Joanna Gamble-George presenting. Dr. Gamble-George is a health scientist and AAS Science and Technology Policy Fellow at the National Institutes of Health. She graduated with honors from Xavier University of Louisiana with a double major in biology and biochemistry and a minor in mathematics. She then returned to her native Tampa to complete a Master of Health Administration degree from the College of Public Health at the University of South Florida before going on to earn her PhD in neuroscience from Vanderbilt University. She also completed a postdoctoral fellowship at the renowned McKnight Brain Institute at the University of Florida before accepting her current position at NIH. In her role as a fellow of the American Association for the Advancement of Science, Dr. Gamble-George serves as an expert science advisor for other scientists as well as medical professionals across the nation who are conducting implementation research on mental health and chronic disease. Given her considerable accomplishments, she was selected for the AAS If-Then Ambassador Program, which brings together women in a variety of STEM careers to serve as high-profile role models for middle school girls. Dr. Gamble-George has received notable recognition and many awards for her strong dedication to mentoring the next generation of scientists. Some of these you've read about in the announcement for this talk, but unfortunately, if we recited all of her accomplishments, we wouldn't have time to hear her speak. So you'll have to read up on her yourselves. We're particularly excited to have Dr. Gamble-George with us tonight because her career so clearly demonstrates Mind's fundamental focus on how combining the knowledge and tools from various disciplines will serve to advance us farther, faster, and into areas of discovery that we can only envision if we get out of our lanes and collaborate across fields, or get out of our colleges and collaborate across campus. I think I might have forgotten to mention that she's also the co-founder and the Chief Operations Scientific and Medical Officer of SciEx, which is a biotech company employing wearable technologies and exploring data science-based methods to combat brain disorders and other health issues. Dr. Gamble-George's work brings together basic and applied neuroscience, public health, and advocacy in a way that makes real changes in policies and also in people's lives. So we are thrilled to host her for this talk, Harnessing Brain Science to Shape Healthcare.
Well, thank you so much, LSU, for just having me and giving me the opportunity to present on some of the research I did as a PhD student, as a postdoc in neuroscience. So this evening, I am going to give you a snapshot of my STEM journey and how my life experiences, especially those involving my research work in brain science, helped me to see how I could make an impact improving the healthcare industry. So I was raised in rural Alabama on my grandparents' farm. And as a young girl, my grandparents really instilled in me the importance of acquiring a quality education or knowledge to make better informed decisions in my life. And they also showed me the importance of community service and how it can help an individual build character and find a new passion, and even promote personal growth and mental health through self-esteem and a sense of accomplishment, while also doing good for the community. Also during my upbringing in rural Alabama, I witnessed a lack of medical care and equipment and a need for more primary care physicians, as well as specialists in disadvantaged populations due to poor social determinants of health. And residents often had to drive 30 miles or more to visit a physician for a wide range of ailments. That could range from the common flu to severe complications from diabetes and cardiovascular disease. And my grandparents were among these individuals where their own city could not provide for meeting and serving their healthcare needs. So this experience and the values my grandparents instilled in me compelled me to believe that I could alleviate these impediments to patient care if I pursued a career that could shape the healthcare industry. So I majored in the biological sciences as an undergraduate at the University of Louisiana. And I later attended the University of South Florida College of Public Health to obtain a master's degree in health administration. During graduate school, I participated in a program sponsored by the American Hospital Association, where I served as a health system specialist in a VA hospital. And in this role, I was exposed to health services research and had to come up with solutions to improving poor patient-provider relationships due to understaffing of medical personnel and preventing delays in patients being admitted to medical units as a result of problems with processing a patient's health benefits applications. And this program really gave me firsthand experience to improving the healthcare industry, as I dreamed of doing during my childhood in rural Alabama. However, I still felt that I needed to do more, especially in terms of understanding how poor social determinants of health can impact an individual's health outcomes. So I went on a mission trip and I volunteered with medical, physical therapy, public health, and nursing students in the U.S. Peace Corps in El Yaya, Dominican Republic. And the goal of the volunteer experience was to implement outreach and sustainable health projects for underserved communities in the Dominican Republic. So I got the opportunity to build ventilated pit latrines, and I also provided free antiparasitic medication in a clinic and health education workshops on a variety of topics such as hygiene, water sanitation, oral rehydration, maternal and infant health. We even talked about sexual health and body mechanics and ergonomics to those that were living in a village in El Yaya. And the interesting thing about the residents in that village, that they had very limited access to education, also healthcare, as well as electricity. They also encountered daily issues with hygiene and sanitation due to a lack of clean running water, which was substituted at times with rainwater that they used for bathing, cooking, and drinking. And the residents also endured vaccine preventable and vector-borne diseases. So I think from this experience, I began to realize the significance of connecting community-based and patient-directed nonprofit and even government agencies to medically underserved communities. And these organizations, what they can do is that they can deliver comprehensive, high-quality primary healthcare services to the most vulnerable populations. That could be people that are homeless, agricultural workers, public housing residents, even combat veterans, and integrate access to health services in areas where there is economic, geographic, and even cultural barriers that limit access to affordable healthcare services. To gain more exposure to health disparities and equities and how I could implement projects to combat these public health concerns, I served on an interdisciplinary team of health professionals at a community center to develop and implement community-based projects in a rural underserved community in Florida. And there I helped with identifying many barriers to healthcare in that community, including but not limited to financial insecurity, health illiteracy, and problems with accessing certain healthcare services. So as a solution to addressing these barriers, I helped with implementing health education interventions at the community center for preteens and teens. So I was able to provide instruction so up to about 40 teens aged 12 to 16 during a teen empowerment program on cardiovascular disease, diabetes, HIV, infection, AIDS, as well as dental health. And I also helped with developing and implementing a healthy nutrition program for up to about 100 camp youth that were aged 5 to 12. So as part of the health education curriculum for the preteens, we invited guest speakers to discuss diabetes and how to prevent the disease. We also held demonstrations on how to measure one's blood sugar levels to help manage diabetes and prevent further complications. We as well did interactive activities with the preteens to promote cardiovascular health, such as teaching them how to check their heart rate at their risk. So for topics such as HIV/AIDS, nutrition, and physical activity, The activities we implemented were more group-oriented and interactive. So for example, we had the preteens participate in food group relay races, where they had to match a food product with the correct food group in a race format. Also, to address some of the barriers to healthcare dealing with health insurance plans, we informed the preteens about how to enroll in a health insurance plan called Florida Kid Care to obtain affordable health benefits. And we also provided a flyer on the health insurance plan for them and their parents. In respect to dental health, we gave a lecture on how to maintain good dental health and handouts to reinforce what we taught, such as how to floss and brush your teeth properly. For the teens, we held very similar activities as with the preteens for diabetes and cardiovascular health. However, we also utilize visualizations of the topics in the form of video, so just help with reinforcing the importance of these health topics. And we also implemented a reward system if the teens completed the in-class assignments we gave them to sort of help with motivating them and promoting good behavior, as well as stimulating their learning. So for health topics such as HIV/AIDS, nutrition, and physical activity, we brought in people that were living with HIV/AIDS and people that were really involved with promoting nutrition and physical activity, just to talk to the teens about how their lives have been impacted by HIV/AIDS, as well as getting good exercise. We also gave quizzes on the health topics and held a potluck with healthy food choices and provided a recipe for a healthy snack such as a yogurt parfait. As with the preteens, we also provided information to the teens about health insurance plans and how they could contact a lot of healthcare organizations that were in their surrounding community where they can obtain service for various medical concerns. To determine the barriers and the facilitators of implementing the educational interventions, we conducted what is called a SWOT analysis, where we were looking at the strengths, weaknesses, opportunities, and threats. And we found that the staff at the community center were very helpful for the planning and implementation of the project. The community center was also well equipped with a lot of resources to help facilitate our educational activities. The barriers to implementing the project, though, really focus on time constraints. Sometimes we had issues with scheduling some of our activities. Maybe they took a little bit too much time as expected. Also, there was limited resources with respect to the bandwidth of the internet, as well as we had issues with sometimes the weather interfering with some of the outside activities that really focus on promoting physical activity. Also, we found that the healthcare organizations in the surrounding community were very helpful with providing speakers to talk about the health topics we covered in the project. In order to evaluate how successful the project was, we implemented a presentation satisfaction survey to the preteens and teens, and we asked the preteens and teens to rate several statements related to the presentations our team gave to the teens and preteens. And the majority of the ratings for the statements ranged from a level of 4 to 5. So many of them agreed with the statements, and they really enjoyed the quality of our presentations on the various health topics. Some of the characteristics include that we asked them questions about were like the pace of the presentation, maybe their level of interest or engagement of the target audience, also the overall usefulness of the information presented, especially in terms of what was being done with the material that we were giving them in the class after they obtained it. So were they passing it on to family and friends, or utilizing it for themselves to improve their health? So the other part of the satisfaction survey included asking the preteens and teens to determine on a scale from 1, meaning not all important, to 5, meaning extremely important, which health topics that were presented to them by our team were of importance to them. So once the surveys were collected, the results from the scale of importance based on specific health topics were placed in the form of what is called a Q-sort. So a Q-sort just ranks the health topics that were presented to the survey participants on a perceived level of importance. So each health topic was given a score that indicated the survey participants' perception of that health topic's importance. And then for each health topic, the scores were averaged. And a Q-sort method allows surveyors to determine which health topics should be expanded, contracted, or maintained for future use in the form of presentation. So as presented in the results of the Q-sort for the teens, the most important health topic presented was in cardiovascular health. But for the preteens, it was mainly dental health. And then the least important for the preteens was between health insurance and diabetes, but for the teens, it was health insurance and dental health. And we thought maybe the reasoning behind some of this was that I think for the preteens, the dental health, some of the topics had already been discussed as part of the program with other staff members. And also some of the preteens were already enrolled in affordable health insurance plans as opposed to the teens. Some of the teenagers, they didn't rate some of the topics as important because they didn't always understand how important the topic was to their health when it comes to like dental health. So what we thought with these topics that they didn't rate as important, we will actually expand on them. So one recommendation was to expand HIV health topic and maybe maintain the remaining 4 topics for the teenagers. And another recommendation we came up with was to include nutrition and physical activity in the list of health topics. So our team should be able to present this to the preteens and teens. And several of the other topics, such as diabetes and cardiovascular disease or heart health, which our team presented to the preteens and teens, are actually linked to nutrition and physical activity. So that's why we want to stress that more. So the projects I mentioned earlier focus on health disparities. When I was completing them, I also volunteered in the VA hospital with different, sort of implementing different fun activities for veterans that suffered from PTSD. And also these veterans, they had poor management of their medical condition. So as a result, I developed this sort of this curiosity about what I could do from a scientific perspective to understand PTSD and find a possible solution. So this volunteer experience led me to pursue a PhD in neuroscience at Vanderbilt University, where I got to study the endocannabinoid system. So you're probably wondering, what are endocannabinoids? So endocannabinoids also are called endogenous cannabinoids, and they're these lipid-derived molecules that activate endogenous cannabinoid receptors in the brain. And the receptor, I will be talking a lot for the rest of my talk. It's going to focus on the cannabinoid type 1 receptor. So this receptor is the same receptor that is activated or turned on by the active metabolite of marijuana. Except for us, we're actually trying to find a way where we can create drugs that will target those same receptors. So endocannabinoid signaling can regulate anxiety-like behaviors. And this is important since anxiety disorders are the most common mental illness in the US, and millions of people in any given year are affected by these disorders. So when you activate or block the cannabinoid type 1 receptors, since I did a lot of preclinical animal studies, what an animal will do is that animal will exhibit certain behavioral characteristics. So activating the cannabinoid type 1 receptors with an agonist such as THC, which is the active metabolite of marijuana, will bring about a reduction in anxiety behaviors. Whereas if you block that same receptor, you do the complete opposite. If you do it with a drug that's an antagonist known as rimonabant, it will have the opposite effect on anxiety. It will actually increase your anxiety levels. So one of our major findings in the lab was that blocking the cannabinoid type 1 receptors can elicit behavioral responses linked to anxiety only when an animal is subjected to a stressful external stimuli. And in this case, we would use a novel cage that was in a very highly illuminated setting, which actually will increase anxiety levels in animals. And the animals that I will be focusing on are rodents or mice. So we believe that we saw these type of behavioral characteristics in our animals um, only when they were treated with the blocker of the cannabinoid type 1 receptor, known as rimonabant. And it sort of relates to this theory that endocannabinoids are synthesized, or the cannabinoid receptor is modulated only under stressful conditions. So synthesis or modulation occurs on demand of cannabinoids, or when there's a physiological need to dampen a stress response. So next, we wanted to see the effects of targeting molecules in the endocannabinoid signaling pathway on anxiety-like behaviors that are upstream of the cannabinoid receptor. And the molecule that we targeted was an enzyme that breaks down the endocannabinoid called anandamide. I might refer in the presentation to anandamide as AEA. And anandamide is actually broken down by the enzyme COX-2 or cyclooxygenase 2. So we had this hypothesis that if you inhibit the enzyme COX-2, which is known to break down the endocannabinoid anandamide or AEA, that will reduce stress-induced anxiety-like behaviors in mice by increasing anandamide levels in the brain. And we believe that the mechanism was occurring through glutamatergic neurons, or neurons that produce the neurotransmitter glutamate. So we've recently shown in a past publication in collaboration with another lab at Vanderbilt that COX-2 is an important modulator of endocannabinoid signaling in the brain. And COX-2 is well known for producing prostaglandins from arachidonic acid. However, we've developed actually a new type of what is called a substrate selective inhibitor of COX-2, or what I refer to as SSCI, in collaboration with this other lab group. And we call the drug LM4131. So LM4131 is one of these SSCIs, and it prevents the formation of, but it does not inhibit the oxidation of endocannabinoids. So COX-2 is like this really important regulator of anandamide, and that is why we are studying it as a target for drugs that can possibly be used down the line in humans to help with addressing anxiety disorders or reducing anxiety in people. So this is the behavioral essay I did a lot of my experiments in. It's called the novelty-induced hypophagia essay, and it helps with detecting anxiety-like behaviors in mice. And it spans over 8 days, and it begins with you acclimating the mice during the acclimation period to their home cage, which is just a single house Allentown cage. And then for 3 days, the animal is trained to consume a vanilla shake in the same home cage setting under red light illumination. Next, the animal is given like an IP injection. So we'll give them that COX-2 inhibitor 2 hours before we do some additional testing. And during the testing, we'll actually place them in a well-lit novel cage and then see, because that environment is now stressful, will the drug after 2 hours bring about a reduction in anxiety in the mice? And we observed or measured anxiety by the amount of time it actually took the animal to take the first sip of this sweet consumable shake. So the theory that we have is that when we administer the COX-2 inhibitor, it's supposed to increase anandamide levels in the brain. So we did mass spec in determining that, and we did find that brain anandamide levels were increasing in the brain after we gave or administered the COX-2 inhibitor to the mice. But the interesting thing is that when we actually performed that behavioral test, the NIFS assay, there was actually no changes in anxiety. The animals that were given a vehicle versus the ones that were given the COX-2 inhibitor, there was just no differences. They actually went up and took their first sip of the vanilla shake pretty much around the same time. So they were both not showing any signs of anxiety. But the interesting thing is that when we repeated these same experiments after the animal was subjected to some form of stress, and that form of stress is foot shock stress, and then we also gave them 2 hours before we test them in a novel cage, we found that there was a huge difference between the animals that were treated with the vehicle versus the ones that were treated with the COX-2 inhibitor. So we found that you have to apparently expose the animal to some form of traumatic stress in order for the drug to actually work. So it seemed like it works very well under conditions where the animal is highly stressed. So next we wanted to determine at what time point the drug actually worked. And so we did a lot of time-dependent studies measuring anxiety behaviors in the mice. And we found that the best time point was actually at 8 hours. So we will stress the animal out, give them a foot shock stress, wait 8 hours, and then we will do our novel cage testing to see if there's any differences in anxiety levels, and, after we administer the COX-2 inhibitor. So just to really also prove that our results that we were getting earlier were correct, or we just wanted to verify them, we also perform additional anxiety like behaviors and other behavioral tests. And one of them is the elevated plus maze. Some of you might know what the elevated plus maze, some of you might not. But it's a maze that measures anxiety, and it really takes advantage of the fact that mice gravitate to dark areas to feel safe as opposed to well-lit areas that are open in the environment. So in the elevated plus maze, which is a maze in the form of a plus sign, an animal would consider to have increased anxiety if they spend a lot of time in the closed, dark arms. Also, the opposite is true. They will have less anxiety if they spent more time exploring the open, well-lit arms. So before I get into the last result that really summarizes what I found in my study, I want to go a little bit over some neuroanatomy and talk about the brain region that we focus on when it came to endocannabinoid signaling. So we did a lot of our electrophysiology experiments in the amygdala. And electrophysiology, if you don't know, is a technique that helps you with measuring neuronal activity in the brain. What we did in this paper, we did both ex vivo, which means outside of a living organism, and in vivo. So the next result I'm going to show you actually combines the elevated plus maze with the EFIS simultaneously, known as in vivo electrophysiology. So we focus on the amygdala for several reasons. One is that it plays a role in emotional processing. And this is because it assigns emotional significance to and produces behavioral responses to external stimuli. There are many connections to other brain regions that are involved in emotional responses linked to stress. One of those brain regions is the hypothalamus. In addition, the central nucleus of the amygdala is the major output brain region of the amygdala and plays a role in regulating the fear response. So studies have shown that lesions to the central nucleus of the amygdala, or CEA, can block fear condition responses. So the amygdala is composed of three subdivisions: the lateral nucleus, the basolateral nucleus, and the central nucleus. So the next slide, I'm really going to focus on how we combine that behavioral technique with doing recordings in the amygdala at the same time to see if our drug actually worked and what brain region was responsible for that effect. So this is a result from where we did in vivo electrophysiology. So we had the animal actually, in the elevated plus maze. We had already exposed the animal to stress, and then we, at the same time, while they're exploring the maze, we were also measuring their neuronal activity, and we were looking at excitatory response, because I had mentioned earlier that we believe that the mechanism of action for this drug actually occurs in glutamatergic neurons. And what we found is that as the mice, once we put them in the center of the elevated plus maze, and we wait to see if they're going to go to the open arm that's well lit versus the closed dark arms, we found that when they exited after going into the closed arms and going to the center part to explore, to see, if possibly they can actually venture out into the open arms, it seemed that the mice that were exposed to stress and treated with the COX-2 inhibitor actually went to the center of the elevated plus maze to, explore the open arms more than the vehicle treated mice. So this lets you know that particularly in the amygdala is the brain region that is modulating these anxiety-like behaviors, and the drug is actually modulating that. So it does help with reducing anxiety. So after my PhD, I completed a postdoc fellowship at the University of Florida. And I did something a little bit different. I moved away from anxiety and depression, and I got into drug addiction and HIV infection. And we really wanted to understand the cellular and molecular mechanisms that drive drug-seeking behavior in people that live with HIV or AIDS. And we had this hypothesis where people that are infected with HIV, somehow HIV itself infects not only the neuronal cells, but non-neuronal cells like glia. And we are focusing on microglia. And we believe that specifically the protein HIV-1 TAT is affecting the microglia and leading to this dysregulation of dopamine homeostasis in the brain. And we thought through a series of events, eventually it leads to this low dopamine state such that people do develop HAN or HAD. It stands for HIV-associated neurocognitive disorder or HIV-associated dementia. So if someone has HAN or HAD, and even if they are on antiretroviral therapy or not, we believe that when these people experience stressful events in their environment, they're still going to go out and seek drugs to abuse or misuse. So here is just a picture showing the brain regions that are involved in drug-seeking behavior. We didn't focus on all these different brain regions. We really focused on the midbrain. And specifically, we focus on the ventral tegmental area, the VTA, and the substantia nigra pars compacta. So one of the first questions we wanted to ask, because we were trying to really understand these cellular mechanisms, is how is it that HIV-1 TAT is affecting how microglia is interacting with dopamine neurons. So we really paid attention to interaction of the processes that are coming from microglia with the dopaminergic neurons. So the image to the right, the green represents the dopamine neurons and the red represents the microglia. And we did it into two different brain regions, the SNC and the VTA. And what we found that there really wasn't too much difference between, and these were done in animal studies. So the animals we were working with, we had control animals and we had another set of animals that actually produced HIV-1 TAT after we administered for 7 days doxycycline. So what we found that there was no really major difference between the control wild type animals and the ones that can produce HIV-1 TAT. But we did find out that there are brain region differences between the VTA and the SNC, such that it seems like the dopamine neurons in the SNC do not receive a lot of support from microglia. So the processes that are coming from the microglia don't necessarily interact a lot or come in contact with the dopamine neurons. So then we wanted to see how HIV-1 TAT really expression affects tyrosine hydroxylase, which is an enzyme that plays a role in the biosynthesis of the neurotransmitter dopamine. And what we found that it actually decreased TH protein levels in the SNC, but not the VTA. So then we want to see, okay, so it's decreasing tyrosine hydroxylase or TH levels at the protein level. What about mRNA? Is it doing the same thing? It didn't do actually anything at the mRNA. So we're seeing that the changes that we believe are due to this hypodopamine state really occurs at the protein level. Next, we want to see how it affects neuronal activity. So we actually measured doing whole cell patch clamp, measuring spontaneous firing activity in the midbrain of dopamine neurons. And we found that HIV-1 TAT actually reduces the spontaneous firing activity of these neurons. So after my postdoc, I went to NIH and I did a AAAS Science and Tech Policy Fellowship. So with this fellowship, they place you at different federal agencies and you get to really get involved with doing policy type work. So I'm actually at the National Heart, Lung, and Blood Institute. And there I serve as a science officer where I'm giving advice to other scientists that are working on these clinical trials that are part of what they call the PRECLUDE Consortium. So they work with people that are living with HIV, but they also have these co-occurring chronic heart, lung, blood, and sleep diseases and disorders. And what they're trying to do is try to integrate other services to combat the chronic disease part into the HIV care continuum. And this is really important because people with HIV are living longer with a lot of comorbidities like heart, lung, blood, and sleep diseases and disorders, even though they're on antiretroviral therapy. Also, people with HIV, they're at a higher risk of hypertension as well as cardiovascular events. And it's also common to find people who are HIV positive, but they also suffer from chronic obstructive pulmonary disease. So, as I mentioned before, the existing HIV care continuum doesn't really include guideline-based care to help with managing these chronic diseases. So there's a lot of barriers to actually prescribing preventive drugs to target some of these disorders, just like such as cardiovascular disease. And also the other issue is that it's not really integrated in mental or trauma healthcare. So one of the groups that I'm working with, they're actually trying to integrate services to combat trauma that HIV positive people experience in their lives, in addition to helping with the HIV care continuum. So this is just a map of all the different sites that I serve as a science officer for. So it's 5 different multidisciplinary groups looking at issues with blood pressure, cardiovascular disease risk. Some are getting into behavioral economics and looking at use for a particular prescription that can reduce CVD risk. And then some, like the one I mentioned, also is looking at the integration of mental health with the HIV care continuum. So last I'm gonna go into how I started my biotech company. So during my last year of my PhD, a friend of mine that I knew back at University of South Florida, we decided to put our 2 expertise together and really try to make a social impact. And one of the first public health problems we wanted to target focuses on heart disease. So there are a lot of people every year that suffer from heart disease or that die from heart disease. And there's about a little bit over 600,000 people that die each year. So that's basically about 1 in every 4 deaths can be contributed to heart disease. So we thought that this is a major public health issue. And the other thing is we looked at it as a passion project from the perspective that on both of our family sides, we had people who died due to heart disease or suffer a stroke or a heart attack, or even have heart failure. So a solution that we came up with was to create a wearable device alert system that has 3 main functions. One, that it can monitor your heart health by taking data from a wearable device, as well as also taking pre-existing medical history, combining that to make sense of the data such that it can predict your medical state. And sort of just make inferences in order to prevent you from getting to the hospital in time and having the EMS to actually come to you so they can take you to your heart health team at a hospital before you actually have a heart attack or stroke. So our main goal is to prevent the number or reduce the number of deaths due to heart disease. And we're hoping to really take advantage of blockchain technology and machine learning to do this. So you're probably wondering, so where am I going with all of this? I've done research in health disparities. I've done a little bit of brain science work. How does this all come together and what I really want to do for the long term? So my next step in my life is to go back to academia. I was doing the AAA Science and Tech Policy Fellowship because I wanted to get exposure to how, as a scientist, I can integrate that into policy work and also use the experiences from my fellowship to one day serve on a federal mandated committee where I can advise policy from a scientific perspective. And I think that's really important because I think there's a lack of scientists that are able to be involved with policy work and communicate our science to a lot of these policymakers like congressmen. I think if we can able communicate our science to them, then maybe we can help with furthering scientific discovery and helping with increasing research funding to also stimulate and promote scientific discovery. So what do I want to do when I go back to academia? So that is my ultimate goal, to establish my own independent lab. I am looking to hopefully examine how interactions between social factors, so that's like social interactions between people, social capital. Also looking at social support systems, how lifestyle factors, environmental factors that can deal with pollution, genetic factors, cellular, and even molecular factors contribute to brain disorders such as anxiety and depression. And then I want to be able to monitor these interactions with current technologies that are in place, or maybe develop new technology to monitor them. And then next, I want to be able to predict when these interactions can cause adverse clinical outcomes. And the last thing I would like to do, the next step, is to help manage and prevent these interactions from contributing to the development, progression, even the severity of brain disorders. And the brain disorders I'm particularly interested in are the ones that focus on anxiety and depression. So that is the end of my talk. Do you have any questions?
Hi. So people can't unmute themselves, so I'll do all the clapping. That was great.
Okay.
Thank you so much. So I have some questions.
Okay.
That was really, it did, your work brings together, you know, it's really interesting to travel on your journey kind of with you through all of these different kinds of, but, you know, interrelated areas. But so one question I have is just that, you know, we're you've done a lot of work with anxiety in an animal model. And I'm just wondering, what's a healthy way that we can stimulate our endocannabinoid system? Did I say that wrong?
No, you said it right.
Endocannabinoid. Sorry.
Healthy way. I could say one example is possibly doing things, because usually, well, let me just step back. So the way the endocannabinoid system works, the endocannabinoids are usually stimulated only when you're under stressful conditions. So you're not going to always have a stimulation when you're just relaxed and you're okay and you're feeling well. It's usually when you're in a state of disease or have some symptoms dealing with anxiety. So a way to, I would say, let's say there's some type of dysfunction with the endocannabinoid system, because there are genetic variants of the endocannabinoid system, especially some of the enzymes that are involved with it in people. So let's say you're one of those individuals where it doesn't work as well or as properly. I would say the things you can do to stimulate it would be to do things that really help with reducing your stress levels, do things that bring you happiness in your life. There are times people will get involved with what is called mindfulness. And those are things that will help you to sort of just take a step back from all the things in your environment that's causing you to stress, and just to focus on something that brings happiness and relaxation. For me, that would be drawing and painting as an example. So I'll focus on that. I'll get really into my drawing and painting, and without thinking about the things that are bothering me. It'll just help to get you calm down. Now that won't help to solve the problem at hand. There's other techniques you can do for doing that. Definitely getting help, outside help. If you feel like it's so uncontrolled that you cannot deal with it yourself. So go talk to a clinical psychologist. If your problems are associated with social economic issues or any other type of social issues, you can always talk to social workers that can give you advice and connect you with services to solve those problems. So for me, when it comes to dealing with anxiety, I always question what is really causing the anxiety. And I try to, sometimes I do that mindfulness to step back to sort of clear my mind so that later on I can think about it thoroughly and break it down to try to address the issue at hand.
Okay, awesome. And I see that we have a couple of raised hands. So Rebecca Wickens, I'm going to unmute you so you can ask your question. Oh, NBC, if you want to turn on your video.
Yes, just give me 1 second. I'm going to try to connect my video. I'm not sure how to do so. So I am calling you guys.
When I was also a PhD student, I applied for this, to attend this meeting over in Ladau, Germany, where I had to compete at 3 different levels, at my school level, at the national level, and the global level, against other young scientists. So these are PhD students, postdocs, and even junior faculty in my field. And I beat out, I think, 19,400 people, because 600 of us were selected to go and meet with Nobel laureates to talk about research in the perspective and looking at it, how can it help with medicine or advancing the field of medicine. So a lot of great things happen during my PhD program, but it's not just intellectual curiosity that will help you advance in your PhD program. It's also how you apply yourself It's also how you market yourself. It's also how you network with people. Networking is very important. I didn't feel like I had a big network during my PhD, but I did have good mentors and a support system where I could at times sometimes go to people and ask for advice about my grant proposal that I was putting in to maybe help with funding future years of my stipend and the research I was doing in the lab. Sometimes I can go talk to people about personal things because, you know, we do have to try to have a work-life balance when we're in the lab. So I will say I definitely made the most out of my PhD experience. And the other thing that I did in terms of making the most out of it, I did a lot of trainings, took a lot of courses, professional development courses that were offered at Vanderbilt and sometimes outside of Vanderbilt that would help me with meeting my professional and career goals once I left my PhD program. I'm always the type of person I try to plan in advance. I look at what I want to do 5 years, 10 years, or so on and so on. And I write down, okay, I want to take this course. I want to get this training. I want to talk to this person. I want to do this informational interview with somebody so they can tell me about their journey and see what steps I need to take. I just make a plan. And I would have to say, when it comes to your journey, it's not a straight path. There's a lot of twists and turns. It has been a lot of twists and turns for myself, but I've enjoyed the ride and I realize I am where I'm supposed to be right now in my life. And sometimes you just got to go with the wind and always try to have a contingency plan or a plan B, a plan C when something doesn't work out. I remember when I applied for the AAAS Science and Tech Policy Fellowship, I didn't get in the first time. So I made sure I had another plan in place. So you know what I did? I went and I did another policy program through the Society for Neuroscience. That program helped me to get into the AAAS Science and Tech Policy program the second year, because I was able to get a letter of recommendation from the person who was over directing the program. So the thing is always try to make a plan and put it in place, but know that, hey, you have to have some wiggle room and some flexibility with that plan.
Thank you so much.
That was a great question, and really interesting to hear more about, you know, how you've ended up where you are today, right? I wanted to ask about, actually, I wanted to mention, since you mentioned that you had presented to Nobel laureates, I looked you up on Eurotree and I saw that you were also academically descended from a Nobel laureate.
Yes, I am.
So the academic great-granddaughter of Eric Kandel.
Yes.
Very famous neuroscientist. So your work at NIH and at I don't see any other raised hands right now, so just a reminder that folks can raise their hands if they have any questions. Oh, okay, so I do see a raised hand now, so I'm going to save my question for a moment.
Okay.
I see 2 raised hands, so. This is Dr. Wallace?
Oh, yes. Hi, I'm Gloria Jean Wallace, and I'm a professor at Xavier. So this is really nice. And when I heard about you, I invited a lot of people. So hopefully there are a lot of other Xavierites on here, including some of my graduate students.
Great.
I'm a medical speech language pathologist. And first of all, I really enjoyed this. This was fabulous because you went from the very highly technical to the just down there explaining what the real issues are and then showing how you're bridging. So that was really beautiful and very enjoyable. So I'm a medical speech pathologist, and I think there's some others of us on here. And I'm just curious, my specialty is stroke and aphasia, a language impairment, you know, after stroke. So I was really excited to hear that your work will be applied to folks who have heart attacks and strokes and that sort of thing. That's my passion as well. And I'm wondering, could you talk a little bit, since you're really into the stress factor, could you talk just a little bit about the role of poorly controlled stress as a contributor to, you know, stroke and heart attack and that sort of a thing? Thank you.
Yes. So there has been a lot of studies where they're now looking at an association between especially chronic stress and increased cardiovascular disease risk. So there's one study where they actually found that people who experience chronic stress, they down the line will experience coronary heart disease. And some of them believe that that association is actually due to some form of increased inflammatory response due to the chronic stress that they're actually enduring. So apparently with inflammatory responses, if things are not currently functional, and also you have to think about, people have already these underlying medical conditions where the inflammatory response cannot be dampened, which means that over time, the negative effects of the chronic stress will get worse and worse on the patient health outcomes. And that will lead to a more increased risk of CVD. And from CVD, that's also linked to these life-altering or life-threatening events like heart attack and stroke that will happen down the line. So I know a lot of people are trying to really understand from a molecular level what is really going on and how there's a connection between the brain and the heart, because the nervous system, it controls regulation of your heartbeat. But is it that maybe the inflammatory responses, because they sort of bring out such a dysregulation, that maybe that's leading to these irregular heart rhythms, and down the line that increases your CVD risk, and later on you can get like a heart attack or stroke. So people are trying to really understand like communication between the central nervous system and the cardiovascular system, and really what happens when someone is under chronic stress. I haven't seen too many. I've seen a few studies talk about acute stress and CVD risk, but it seems like a lot of them are saying you have to be under this like constant, continuous, or exposed continuously to stress for it to really lead to some life-threatening health outcomes with regards to cardiovascular disease. But I think a big issue is that the stress itself causes heightened level of inflammation. And that sort of also relates to what I was doing when I was talking about the drug addiction and the HIV interactions and looking at the same molecular mechanisms. Also, that is dealing with an inflammatory response, because microglia, they release things out into the brain that negatively can affect the neurons and bring about neuroinflammation. So it could be that that is one of the mechanisms that is actually occurring. Maybe the glia that's in our brain, maybe there's things that we're, experiencing in our external environment causing the stress. It also could be as well what we're eating too, because there has been links between, communication between your gut and your brain. So it could be that maybe the food or the diet and lifestyle you're living is also causing this inflammatory response. But maybe the inflammatory response is coming from the dysregulation within the non-neuronal cells, and then those cells, whatever they're releasing into the environment, are negatively impacting the neurons, and maybe those are the neurons that actually regulate the heartbeat, and that's what's going on. But that's just a theory of mine.
Very interesting. Thank you so much.
And we have another raised hand. Oh, we have another raised hand. So Anna Long.
Yes, thank you so much for your presentation today. I'm actually a professor in school psychology, and I'm a licensed psychologist, and I work with children and youth. And so my question for you just relates to, similar maybe to the last question in terms of your trajectory. I very much have been influenced in my research by sort of real world problems that I see within my own ethnic minority community or other communities of color or low income populations. And I think a challenge, at least for me as an academic, is academia doesn't really incentivize service work so much. And so kind of balancing, being of service to your community, being in this privileged position, know, really high productivity things that academia is. So I'm interested in how you've, you know, tried to like not overextend yourself beyond what's humanly possible.
So you're getting at like definitely having like a good work-life balance, especially when you want to do community outreach and also dealing with, you know, maintaining your publication record, you know, in academia and funding and managing your lab. For me, when it comes to taking on a lot of things, I think initially I was one of those people I always kept saying yes. I've learned now to learn to say no to some things. And I think the other thing is learning to pay attention to your bodily cues, let you know when you're just overburdened and overwhelmed. And just listen to those cues. And I always do what is like a mental health break, which also helps me with just recuperating and thinking things differently, and just recovering from burnout at times. But for juggling multiple tasks at one time, I do a lot of prioritization, where I look at the importance of, you know, what I want to do, especially with respect to how it helped me to advance personally and professionally. So that helps me with determining, okay, should I do this then or should I not do this? The other thing when it comes to doing community outreach and also keeping up with the responsibilities as a scientist with your own lab, I think you have to sort of find creative ways. If you feel like the demands of your work is very, takes a lot out of your time and you might not have that much time for community outreach. There are ways where I've seen some scientists where they would use community outreach and turn it into like a research project. So let's say you're interested in understanding what strategies would help with students possibly pursuing a particular STEM discipline if they come from a disadvantaged background. What are maybe their attitudes toward those STEM disciplines? You could turn it into community service as well as research, where you are setting up an academy or some type of summer program to help with recruitment or to help with sort of reducing any negative perceptions of STEM fields in disadvantaged populations or in people that just don't really know a lot about the field. So maybe that's why they never thought about pursuing it. So in there, by doing that, you're doing community service, but you're also going to do research on it to see if your strategy actually works. So that is one possible solution. The other thing is, I know sometimes I think with faculty members, they do require commitments to serve on committees. So maybe you can work on a committee that focuses on that area of community outreach that you want to do. Or you can find a way of incorporating community outreach with your whole lab, where y'all do an activity together, and you actually go out into the community and do service. I know at my current position, sometimes before the pandemic hit, we would actually go out together and do community service together. So in that way, we're all doing it at the same time. So no one will feel like one is doing more work or overburdened because we're all taking off time to actually do that. So those are just some suggestions.
Thank you.
And we also have a question in the Q&A, which is, are there any areas of neuroscience that you stray away from?
Stray away. That's a hard question. It's a good question, but so I never looked at it from that perspective. I would say that I would possibly initially stray away from a subject if I really didn't understand it well. And then I would come back at it with a strategy to try to improve my understanding. I don't know of any particular areas I have actually done that. So I've always had a curious mind where I always wanted to learn more and understand things, even when I couldn't understand things. And I always like to challenge myself when I don't understand things well to make sure I have a better understanding of them. So it's hard for me to think of any particular subject of neuroscience I stayed away from. I can definitely talk about the areas I would like to get into that I haven't had the opportunity. I know I haven't had too much opportunity to do a lot of human studies with type of brain imaging. I haven't done too many behavioral experiments where you do self-administration regarding like dealing with, you know, when you're looking at like experiments involving drug addiction or concerning that or focusing on that. So I know I haven't done a lot of that. So I would say maybe those are maybe the areas I sort of have strayed away because I just don't have experience doing them. I don't know a lot about them. So I probably would like reach out to people who are experts in that field and ask them for advice and help.
And that was a question from an anonymous attendee in the Q&A. So I don't know if the anonymous attendee wants to, maybe they had something in mind that, you know,
okay,
they were thinking, I don't know. So if you want to clarify, anonymous attendee also. So I'm interested in your work at the, and I don't see any hands raised at the moment. So please raise if you have a question. Or actually, I do see Dr. Wallace's hand raised. I wasn't sure if you have another question or if it's just still raised, Dr. Wallace.
Still raised. Thank you so much.
Oh, okay.
Well, you know what, actually, I do have one related to my other question about underlying, and the reason I asked you that question was, you know, with all the stuff on the news, it's highlighting all the racial kinds of issues that, that Blacks in this country deal with chronically, because you mentioned chronic.
Yes.
So do you have any comment on that as related to some of your earlier comments related to chronic stress, not acute, but the chronic stress?
Oh, chronic stress.
We just become used to it.
So I think you're referring to a lot of the health conditions, I think, with respect to the COVID-19 that are coming out in the open, but they've always been there. Is that what you're referring to?
No. So I'm back to stroke.
Oh, stroke. Okay, stroke. Okay.
Everything is stroke, right? That's my. But so I'm wondering, you know, the thought is that people forget. A lot of Blacks forget that they're under a lot of stress, and so you know.
Oh, okay. Yeah. Oh.
All these things that are disproportionately impacting. So I'm wondering, not just in terms of stress and heart, but stress and heart in Blacks. Any comment that way with chronic?
Excuse me. Mm-hmm. So, yeah, so with African Americans or Blacks, we are definitely affected disproportionately when it comes to cardiovascular disease compared to other ethnic and racial minorities or other ethnicity and races. And a lot of that is because we have, I would say, just to go back into history, we're still living in a country where we're still facing oppression, even though slavery ended hundreds and hundreds of years ago. We're still dealing with not having access to proper resources to just help us with, you know, managing our heart or things that we can be doing to improve our chances of just having, you know, better health outcomes. And also a lot of us live in poor communities that sometimes are near like plants, abandoned plants, where it can lead to pollution and that can affect our health. Sometimes we are in environments where we don't have clean running water. Sometimes our housing might be blighted are not proper, there's a lot of social determinants of health that we are affected by. And then a lot of us suffer from high unemployment rates. We suffer, and sometimes that's due to not having access to opportunity because of racism or biases based off our skin complexion. So it also gets into colorism as well. So because of those biases, sometimes they're implicit, other times they can be explicit biases. And sometimes those biases are actually held by healthcare providers. So because of that, we have poor patient-provider relationships. Sometimes we don't trust the providers as a result. And then there are times when those biases are, or we're subjected to those biases by healthcare providers, we do not get healthcare that we need. I was talking to someone earlier about a commentary that I wrote in regard to this paper where they were talking about how there's a difference in terms of health outcomes, specifically heart health, in Black patients versus white patients. So apparently the Black patient, white patient were going to the same medical or academic medical center. And yet the Black patients, when they're presenting with this increased risk of CVD or having heart failure, they were being serviced by a general service medical unit instead of the cardiology service medical unit. So right there, you know, there's something wrong. There definitely has to be some implicit biases going on. There could be issues with the way they're processing these patients when they come in. Maybe they're not taking them as seriously when people's like, you know, I'm suffering for something. Or it could be that because there's so much that we deal with on a day-to-day basis when it comes to stress, that we have a lot of underlying medical conditions, and maybe our symptomology could be a little bit more complex, but still I would think that even in those cases, there should be a way to streamline it and send the people to the proper service so they get the proper medical treatment. So a lot of the chronic stress just deals with just continuous oppression, not having the opportunity to really have a better life, not living in environments or built environments that are safe for us, that have places nearby that has healthy eating choices. There have been studies where they've talked about how a lot of Black neighborhoods are much closer to stores that sell alcohol versus stores that sell healthy food or grocery stores. Or you might have more fast food chain restaurants in Black neighborhoods, predominantly Black neighborhoods, as opposed to neighborhoods that are predominantly white. So a lot of this, the issues are on so many different levels. So I think that's why I'm really interested in understanding all the interactions from social, lifestyle, genetics as well. So I think definitely in Black communities, we don't always have the support system and services in place, or if they're in place, we don't have the ability to access them properly, or even know how to really make use of them to help with bettering our health. And then there's definitely a lot of different forces that come into play that prevent us from doing so.
Thank you.
So I'm really glad that Dr. Wallace, you brought that up, and Dr. Wallace and I are both in a field in which there's a real issue of representation that I think that we're really going to be hopefully trying to address. It takes a lot for, I'll just say as a white person, to recognize what the issues are and to, make an action plan. And, the issue of representation is so enormous that, I've read studies about how when Black children have Black pediatricians, they actually have better survival rates. So that's insanely serious. And then, when Black men see Black doctors, that they're more likely to opt for preventative healthcare procedures. So I do think it's really important to have those conversations, and it certainly relates to your work in public health. So thank you. don't know if there are any other, oh, I see a hand raised. Are you there?
Hello? Yes?
Yes, you're on.
Excellent. Hi, nice to meet you again.
Yes.
So I don't know much about mice and animal research. I did some when I was doing my master's, but apart from that, I never worked really with mice. It seemed like that you were equating anxiety to reduction in motility. So they were showing, I guess, some freezing behavior and so on. That reminded me to, in Parkinson's disease, if there is a reduction in dopamine, very often you see these individuals have anxiety much higher than the general population, but they also have higher incidence of depression. And in humans, it's pretty easy to ask if this is actually part of their anxiety or if this is part of their depression, that they are not moving. However, in mice, we assume cannot ask. So I'm wondering, how do you know that it is not an effect of depression and it is an effect actually of anxiety for that stress response?
Okay, so I didn't show all the data. We did do behavioral tests that actually measure anxiety. So the behavioral test I was referring to or talking a lot about is referred to as NIST. So there's two components of that test. I just focus on the feeding latency part, and that's related to the anxiety part. But we also would measure the amount of shake that was consumed by the mice, and we relate that to motivation levels or anhedonia. So we also found that the drug can reduce anhedonia, or loss of motivation or hopelessness. But I think it's a little bit more complex, because when we did tests for depression, like we did force, swim tests, and we also did the tail suspension test. So both of these tests are for measuring depressive behaviors in the mice. And we got a lot of mixed results. We were only able to get, I think, one-time significance, like in the force swim test when we did chronic stress in the mice. But with the tail suspension test, it was we didn't see any significance. So it might be that when it comes to depression and trying to understand it in animals, it's a little bit very complex and it's not always concrete. So you have to do multiple tests to determine if the drug is good at that. So I think for us, the next step is try to do it in other animals, or maybe work with a transgenic mouse model that sort of exhibits depressive symptoms, and then see if the drug will still work by reducing the depressive behaviors in those mice that are already prone to it by changes in their, genetic composition. So that might be one way. The other thing is, if it doesn't work, then we would probably have to maybe get to the point where we want to test this in humans. But of course, before you can get to that point, you have to get approval from the FDA and make sure that the drug is safe, and see maybe it's something that we might not see the result in a particular animal, but we may see changes with depression or motivation in human populations. So that might be the other thing. And then another thing I have to mention, when it comes to these behavioral tests, they're a little bit tricky. And what I found when I was a PhD student is that a lot of times, like with us as humans, we have a lot of individual differences. And because of those individual differences, we will cope differently to stress. We don't have the same response.
Absolutely.
The interesting thing, though, I found that in mice. So at one point before I got to stress in the mice and then studying anxiety after they've been exposed to stress, I was just doing control animals, and we kept getting all these variations. So I remember I just went to my PhD mentor and I was like, I got to figure out what's going on. Why is this? So I proposed modifying the behavioral test such that the same test I was using for NIFS, where you can actually measure individual differences. And we modified it such that we found that some mice that when we characterize them by putting them in a behavioral test, and we characterize them as either having already at basal levels low anxiety or high anxiety. And then we would expose them to stress and give them a drug and then measure them again in the same cohort of mice to see if they have anxiety. And we found that sometimes the variations with the way drugs work is due to because there's individual variations within us. How do we deal with stress? Also, you have to think about the environment when working with animals. When you have them in a cage, a lot of animals come in a cage with more than one animal. So sometimes the animals in the cage will get along and other times they fight a whole lot. That is also a level of extra stress. Think about humans. If we're around, violence, it can stress us out. Some of us might be able to deal with it and it doesn't faze us and we go about our day, others can't. So we really have to pay attention to that as well in terms of why we get these different responses.
Thank you. That was a very, very elaborate answer. I really appreciate that. And yeah, one thing that you could do is maybe MPTP mice, the neurotoxin, knocks out the dopaminergic cells and see what kind of effects that has. But,
that'd be very interesting, yes.
Thank you.
And I think we can probably all empathize with your mice in a cage fighting in 2020, right? But I think that actually brings us to the end of our time tonight. So again, thank you so much for your visit, Dr. Gamble-George. It's been a real pleasure. And I particularly enjoyed the Q&A. And thank you to everyone who's been present.
Thank you so much for having me. I really enjoyed all the meetings with the faculty members and talking with everybody. And I love the research that y'all are doing at LSU.
Thanks. And we'll look forward to seeing more of yours too. Yeah. And maybe I'll let